For decades, inherited blindness, especially from a condition called retinitis pigmentosa, or RP, it felt like staring at a locked door. We knew the problem was genetic, right? But the full picture was always just out of reach. Well, in just the last few years, a wave of new discoveries hasn't just found the key. It's kicked that door wide open. So today, we're going to unlock this story and dive right into a revolution that is changing absolutely everything.
I mean, think about that for a second. What if the answer was right there, but we were all looking in the wrong direction? It's a heck of a thought, isn't it? For so many patients and researchers, the answer to that question has changed the entire game, because it turns out the fundamental problem, well, it wasn't what anyone thought it was, not by a long shot. It was so much more complex than just a simple mistake in our genetic code.
Yeah, for the longest time, the main idea, the only idea really, was this. RP is caused by what's called a point mutation. Just think of it like a single typo in the massive instruction manual of your DNA. One wrong letter in the recipe that ends up creating a broken protein. It made sense, it was logical, but as we now know, it was a deeply, deeply incomplete picture of what was actually going on.
Okay, so let's get into the first big turning point, because this is where everything starts to change. The year is 2024, and a new idea just completely shifts the landscape. It turns out the problem wasn't always a broken recipe. Sometimes it was about the cell making way, way too much of a perfect one.
And this really lays it out perfectly. The whole way of thinking had to flip. We went from obsessing over the quality of a gene, is the recipe broken, to looking at its quantity. How many copies of that recipe are there? This brand new way of looking at it is called copy number variation. And believe me, it was an absolute game changer.
So here's how it actually works. It's like this crazy chain reaction. What researchers found was that in some people, the RHO gene, that's the one that makes a super important protein for vision called rhodopsin, it gets accidentally copied. So now the poor photoreceptor cell is just flooded with this protein. It's a perfect protein, nothing wrong with it, but there's just too much of it. The cell's machinery goes into overdrive, it gets completely overwhelmed, and it just shuts down. It literally works itself to death, causing those vital cells to die off.
Okay, so that first discovery was huge, right? But it also raised a bunch of new questions. And it turns out the next piece of the puzzle was hiding in plain sight, in something that's totally unique to us and our primate cousins.
So for years, decades, scientists were using animal models, mostly mice, to study this stuff. And for some types of RP, they just kept hitting a brick wall, over and over. The things that should have worked in mice just didn't work in humans. And nobody could figure out why. What were they missing? Well, the answer to that question was the second huge breakthrough.
And there it is. The answer was a ghost in the machine, a gene that mice just don't have, but we do. All primates do. It's called TRIM49. And suddenly, it all made sense. All those failed experiments, all those dead ends, it's because they were trying to fix a problem in a model that was missing a critical piece of the puzzle. It's like trying to fix a car engine without knowing about the spark plugs.
So what exactly does this special primate gene do? Well, think of TRIM49 as the supervisor for your retina's cleanup crew. It's the boss that tells the cell how to take out the trash, all the waste products that build up. But if that supervisor's asleep on the job, if the gene is faulty, the garbage just piles up and up. The whole system becomes toxic and the cells start to die. It's that critical.
All right, so now we have these two massive turning points, right? The gene overload problem and the primate clue. Once those were understood, it was like the floodgates just burst open. It didn't just solve old mysteries, it gave researchers a brand new map for this disease, and things started moving incredibly fast.
Just look at this timeline. It is an absolute explosion of knowledge. It's incredible. After the big RHO gene dosage discovery in 2024, boom, we get the IDH3 gene, which is tied to the retina's massive energy crisis. Then boom, the TRIM49 cleanup crew is officially discovered. And then by 2026, we find ZNF124, which acts like a master commander for the whole genetic network. In just a couple of years, we went from staring at one broken piece to finally seeing the entire interconnected system.
So, you're probably wondering, how on earth did they figure all this out so quickly? And that's a great question. The answer isn't just brilliant minds, though there were plenty of those. It was also a revolutionary piece of tech that let them see these uniquely human diseases in a way they never, ever could before.
Get ready for this because it's straight out of science fiction. The tech is called retinal organoids.
Basically, scientists can take stem cells from a patient and grow mini retinas right there in a lab dish. And we're not just talking about a clump of cells. These are tiny, complex structures that grow and develop just like a human retina, with all the same genetics, including those primate-specific genes like TRIM49. This is how they finally leapfrogged the whole animal model problem. They could study the human disease in human tissue for the very first time.
So what's the result of all this new science, this new tech? Well, this number says it all. The list of genes we know cause retinitis pigmentosa has exploded to over 100. Think about that. Every single one of those is a new answer for a family, a new target for researchers to aim at, and a whole lot of new hope for people who never even had a specific diagnosis before.
But really, it's not just about counting genes. That's not the main point. The real revolution here is understanding that this is all one big, interconnected system. We finally moved past that old one gene, one problem idea. Now we can see RP for what it is, a systems failure. It could be a breakdown in protein dosage, a crisis in the energy supply, a failure of the waste cleanup crew, or a problem with the command and control network. And knowing that changes everything about how you design a treatment.
So this brings us to the big question, doesn't it? With a blueprint this clear, are we actually on the verge of conquering inherited blindness? You know, for the very first time, the genetic code for this disease feels less like a deep, dark mystery and more like a map, a roadmap. The routes are being charted, the destinations are being identified, and the vehicles to test the journey are literally growing in lab dishes as we speak. We're not at the finish line, not yet. But for the first time in human history, that finish line, it's no longer a mirage. It's finally, truly coming into view.